I read that if just a single parasite makes it to the liver, it can cause an infection. Is that true?
Yeah, that's right. And I think that's the really tough challenge of malaria.
The reality of this problem is the hardness of it is set by nature. And nature is a vicious test setter.
Adrian asked if I wanted to stay on. This was the PhD project that he pitched to me. I thought it sounded quite interesting to make a new vaccine. There was a lot of iteration. The first time I saw the particles under electron microscope, that was really exciting.
Would you participate in the challenge trial?
I would love to, but I really hate needles. I lie down. Otherwise, I might faint. Kind of ironic for a vaccine developer.
There are people like Katharine Collins who invent entirely new vaccines that are now going to be used by millions of children. You may know one of these people. You may become one of these people in the future.
Malaria kills around 600,000 people a year. Most of them are young children in sub-Saharan Africa. It's caused by a parasite, not a bacterium or virus, and it's spread by a mosquito. Until recently, the only control measures were insecticides and a handful of anti-malarial drugs. But in the last few years, we finally got effective vaccines.
2 malaria vaccines have now received WHO recommendations and are being rolled out across Africa. And for the first time ever, a human-infecting parasite has a vaccine.
Getting here took longer than it should have. The first malaria vaccine was developed and tested in the 90s, but it spent 23 years in clinical trials and pilot tests before it was licensed and rolled out.
So, in this episode, we're going to cover the science of why malaria is so hard to vaccinate against, how the vaccines actually work, why it took so long, what we can do to speed up the rollout now, and what even better vaccines are being tested now for fewer doses and longer durability in the future.
Today's episode is special because we're joined by Katharine Collins, who co-invented the second malaria vaccine during her PhD, and she can tell us what it was actually like from the inside. Welcome to Hard Drugs, hosted by me, Saloni Dattani, and Jacob Trefethen. Okay, so we have some news today, right? Because Jacob, you have started a new job. Could you tell us about what you're doing now?
I'm gainfully employed. I'm working at a foundation, making grants to science still, but from a different vantage point. It's the Open AI Foundation, and we have been getting started in science. So it's kind of exciting, a little bit different than my previous job, which was at Coefficient Giving. And the previous job was quite fun, but you'll have to ask around what my reputation was there. For example, Katharine, what was I like back then?
Jacob, you were the best manager I've ever...
No, we can't include that. No, expulsion. No, and the biggest difficulty of changing jobs is that I don't get to work with some of the wonderful people I worked back at Coefficient Giving with, like Chris Somerville, Ray Kennedy, Heather Youngs, Rafa Dibb, Douglas Chukwu. Oh gosh, so many people, Ashley Meow, and guess who? Katharine Collins. Now, the good news is that Katharine, at least, is here today.
So Katharine is joining us from Coefficient Giving as well, and is a very special guest on today's episode because she co-invented the second malaria vaccine.
Now, we are lucky at Hard Drugs to have listeners from around the world, and that means I am going to start here with some insight into the British psyche for those listening from other places. So we have a guest on today who is an inventor, and she's also British. That means that she's very reticent to take credit for things, even things she invented. So it took a lot of cajoling to get her onto this episode, though we knew if we could pull it off, that our listeners would love it. So I am very grateful for the willingness of our guest to be cajoled. But I should say up front, as part of our ability to pull it off, that there are many other people involved in this story of malaria vaccines and of R21 in particular, who we won't get to have on today, who took forward the vaccine into clinical trials, who did all the work to make sure it could get approved and used by kids around the world. So That's my proviso. That's the trade we had to make with our world famous inventor. With that in mind, enjoy.
So I have a question for both of you, which is, do you have a favorite parasite? What I didn't realize until recently was that fungi and plants can also be parasites. And so a parasite could be a single-celled organism. It could be an animal like a mosquito, vampire bats, hookworms, or it could be fungi like the ringworm fungi, or it could even be a plant like mistletoe is a parasite.
What?
Yeah, mistletoe is a parasite. It attaches to other trees and extracts water and nutrients from them.
Gosh, and here I was. Okay, I think I have two answers. You brought up hookworm. Hookworm's one of my answers. Tapeworm's another one of my answers. I mean, tapeworms, disgusting, obviously, but you just have to be impressed. They get so large.
How large do they get?
Like, I don't know, probably miles long. Joking.
Wow.
They get feet long for sure, and maybe meters. I mean, I just think that's crazy. It's obviously gross. But luckily, hookworms aren't gross at all. All they do is hook onto the side of your gut and suck your blood. They come up, they sort of get up through your foot, sneak, their whole life cycle is insane. They eventually get down into your gut and hook onto it and start sucking your blood. I mean, that's pretty impressive.
Is that why they're called hookworms? Because they hook onto your gut?
You know, I've never thought of that. And the answer is probably yes, but I don't know.
And then are tapeworms called that because they look like tape?
No, it's actually because on the underside, they are sticky.
Oh,
that's
wow,
a joke.
I never
Sorry,
knew
that's
that.
a joke.
Oh. You tricked me.
First trick of the episode.
Okay, my favorite parasite. Also, well, I'm not impressed by this. I'm just so horrified by it that I have decided it's also my favorite. And it's guinea worm, which is so horrible. And people ingest the worm larvae when they drink contaminated water. And the larvae get into your stomach. They get through your gut. And then they grow into worms that can reach up to 1 meter long. And they kind of crawl through your connective tissue and your joints, and then slowly erupt out of your legs, usually out of the skin near your legs. And the emergence is also very slow. And the way that you remove the worm is by slowly winding the worm around a small stick. And if you go too fast, the worm snaps and dies. And that causes severe inflammation in your body.
That sounds like it was invented by Jigsaw from the series Saw as a patient for someone who was impatient or something. Okay, well, thank you for that.
Wait. The good news is, though, we've almost eradicated it. Isn't that great? So it used to cause like 1 million or more cases per year in the 1980s. And it turns out just cleaning up the water, filtering drinking water, or telling people not to drink from stagnant water can help prevent infections.
Do you know how near eradication we are?
I think there were 10 cases reported in total last year.
Wow.
Wow, that's incredible.
Yeah, so from millions per year to 10. What about you, Catherine?
Yeah, mine's really boring. Mine's the malaria parasite.
Yep.
I can't say anything else after studying it for for 2 decades.
Yeah, it would be really surprising if you said something else.
Yeah, it would kind of be like adultery or something.
In this episode, we're going to talk a lot about the 2 malaria vaccines that have been rolled out. The first one is called RTSS, and it was developed in the 1990s. The second was R21. It was invented by Katharine Collins, who's here with us today, and it was based on the first one. So those are 2 names that we're going to refer to throughout the episode. RTSS, the first malaria vaccine, and R21, the second malaria vaccine. I guess it's easy to remember because the second one has 2 in it, R21. But just in case that is confusing, those are the two names that you've got to remember. RTSS, the first malaria vaccine, and R21, the second. Maybe let's start with how you got into malaria research in the first place. What drew you into the field, or like how did you get into vaccine research?
I had done my undergraduate and my master's degree in basic research and not in global health. I did various projects in different disease areas, but it was really about, trying to understand the fundamentals and different signaling pathways and things like that. So I think one of my projects identified a protein that interacted with another protein in a pathway. And why that was cool. And I loved science, and it's definitely where I was supposed to be. But I didn't see how that was going to have any direct impact on health in the near term. I actually started looking for jobs or how I could have an impact in global health, or how I could transition from science into global health work. And it wasn't really clear. And then this job was advertised at Oxford University to work on malaria vaccine trials. And I thought, well, that's global health. I should just take that job or apply for that job. And that may open doors. And so I did. And, I really loved working on malaria. It was instantly something I was really passionate about, really motivated to work on. And, it's just a fascinating parasite. So the job was a research assistant on malaria vaccine trials. So the Jenner Institute at Oxford, where Adrian Hill's group that I joined, they were testing a number of different candidates in malaria trials. And my job was to help coordinate those trials and to do all the immunology in the background.
And then how did that turn into inventing a new vaccine?
Yes. Well, after being there for about a year, Adrian asked if I wanted to stay on and do a PhD. And the project idea was that Adrian had developed a vaccine that targeted the malaria parasites whilst they were in the liver. It had been shown to protect some people against infection, but it was quite a low level. I think it was 20% efficacy. So the idea was to combine this liver stage vaccine with a vaccine that can also prevent the malaria parasites before they get to the liver, so stop them invading the liver. And the leading vaccine at that time targeted the parasites before they invaded the liver, and this was a circumsporozoite-based vaccine called RTSS, and it was developed by GSK. So he wanted me to work on making a newer version, an updated version of that vaccine.
That's so cool. I feel like so my PhD was very boring in comparison to this. And it makes me think that there's actually...
Saloni, did you not invent a vaccine in your PhD? Well,
I know, but I feel like people should know up front that some PhDs are better than others. And some fields are better than others. So who else was involved in this? Did you have mentors, collaborators, rivals, or enemies?
I worked fairly alone in the lab. I did have a few people that were supporting the work as well. So my PhD supervisors were Adrian Hill and Sarah Gilbert. There were also a couple of postdocs and technicians and PhD students that helped me with various parts of the project over the years. For example, someone actually helped me vaccinate all the mice that we used for the preclinical experiments. And then I did the immunology myself.
And Sarah Gilbert is the inventor of the AstraZeneca vaccine, is that right?
That's right. Yeah.
For COVID. Got it.
Yeah.
And rivals?
Rivals. Well, you're asking the good questions. I guess in the field at the time, there were lots of people trying to develop a malaria vaccine. I guess there was competition between the different groups to see who would get there first. And whilst I was working on it, RTSS hadn't crossed the finish line. But it was obviously way, 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 way further ahead in development. So the expectation was that vaccine would move forward and get approved first. And then R21 may follow.
Right. And by that point, had the RTSS patent already expired?
Yes, actually, that's right. So if I remember correctly, it was due to come off patent. And interestingly, that was essentially the starting place for learning how to make R21. So I reviewed that patent. I looked at how they'd made RTSS. And then I made a plan for how to go about making R21.
Did that have enough information for what was that like?
The RTSS is actually made with a lot of a protein on the surface of the Hep B virus. This is the Hep B surface antigen. And that was needed to make it form the virus-like particle but it had quite an excess of Hep B surface antigen. So it's actually a really good Hep B vaccine and not a terribly good malaria vaccine. So my project was to see if we could remove some of that Hep B and actually make it a better malaria vaccine. And we had established a plan for how we're going to get there. So initially we wanted to just replicate the RTSS process in the lab and then try a few different things to see if we could get the particle to form without this excess Hep B surface antigen. And so one approach was actually to just try and chop out some of the malaria protein out of the vaccine and remove some of the T cell epitopes which are in there and a few other bits as well, and see if this smaller protein would be more able to form particles on its own. And the other way was just to try and reduce the amount of extra Hep B that we were adding and see if it could form particles on its own that way by using some of these newer yeast expression technologies to grow the protein and new methods to purify it. So I think RTSS had something like a 12-step purification process, at least in the patent. And that might not be what they used anymore. But I wasn't really interested in trying to set up this 12-step new process in an academic lab. I'm not a biochemist. So I essentially looked at their process to understand what they had done and why, and then developed a purification process using a combination of some of those old methods and some of the newer technologies.
I feel like my immediate question is, why didn't the developers of the RTSS vaccine do that? Like, if there were various steps that didn't need to be included.
Yeah, they probably did. When they first patented what they were doing, it probably had this multi-step process. Maybe they had different they were using lots of different methods, so they patented the whole process. And I'm sure they probably optimized that as they scaled up and improved their manufacturing.
I want to just get more of a sense of what it feels like to be in the lab alone, toying with different steps of purification. And how long did you feel sort of lost versus actually it worked pretty quickly and once you saw in the electron microscope, you're like, oh God, it's actually going to work. Was that pretty quick, or you were there for months, years? You're tinkering. What did the invention process feel like?
Actually, I think I was interviewed by someone a while ago, and my recollection is that it was very quick, that I kind of gave it a go and it worked.
Wow.
And then I thought, is this true? And I went back to my lab notebooks and I had a look through, like the electronic notebooks, and I had a look through, and it wasn't that quick. There was a lot of iteration, a lot of optimization that happened with every single stage. So we were using yeast to express the protein and grow the protein, and there were 4 different strains we were testing. And then for each different strain, we picked multiple different colonies, and I screened all of these different colonies first to find the best expressing colonies, and then I started to try and purify them. And I probably used multiple different techniques until I got something that looked substantial. So it wasn't that quick. So I think, it was a long time ago now. So my memory is a bit optimistic, maybe.
So am I imagining right that it's like you have the instructions-ish from the patent, and then you're trying out different options, like different versions of the yeast, different versions of the purification process, and like at each step trying to optimize which is the best one to go forward with, or something like that? Or is that not the right way to think about it?
Yeah, that's just about right. There are a lot of newer technologies available for purifying proteins and particles. So it's kind of a process of looking at which steps could be replaced by newer technologies and going for something that was more modern and simpler where that was possible. And where there wasn't a newer technology, we were sticking with that old process as well.
So you were at the Jenner Institute, is that right?
That's right.
Do they have just people working on many different diseases and like trying to develop vaccines against all of them? Or what are there some areas that they focus on? Or like, are lots of people doing this during their PhD?
I think developing a vaccine was fairly novel at the time, or quite rare. So not a huge number of people were doing it. There's 3 PhDs I can remember that developed a vaccine during their PhD, and I was one of them. But there may have been others as well. But in terms of the structure at the Jenner Institute, there are different group leaders or PIs that have their focus area. And the Jenner Institute was generally open to having new people join the institute. And if there was space, they did actually join the physical lab space as well. So we had work going on multiple diseases at the same time.
Okay, so you're tinkering. You look under the electron microscope, and then you test something in mice, and it protects them from malaria. Did it feel like a eureka moment at any points of that step, or was it just iteration, feels good, feels a bit better?
No, definitely a few eureka moments. I mean, the first time I saw the particles under electron microscope, that was really exciting, especially when I had used the process that we didn't think was possible. So by removing all the excess Hep B, that was really cool when we could see those. And they look very similar to the Hep B particles. So it was really reassuring that we were doing something right. And then the next push was to move that into a preclinical study to inject mice and see if we get an immune response, and then if we can protect the mice as well. And whilst I was optimizing the process, the yield was really, really low of the particles. And I tried to concentrate it, and it wasn't easy to concentrate. So we ended up injecting an incredibly low dose into the mice. And it was like, well, we might as well. We have the product. Let's just see what it does. And then it protected all the mice. And so that was really exciting. We had gone for this dose that was dramatically lower than any other dose we normally test in the lab of a vaccine. I think 10 times lower than the patent had showed for RTSS being tested in mice. And then it worked. So that was really exciting. That was one of the really exciting moments, yeah.
That's super cool. Because if you have a lower dose, you can vaccinate more people with the same amount, right?
Yeah, exactly. And a 10-fold lower dose, that's huge. That's not half doubling the number of people. That's a dramatic increase. But actually, at the time, there wasn't a lot of information on how well dose in mice would translate to humans, and if it would actually result in a lower dose human vaccine. We did later find out that it was easier to scale up.
It's so interesting how the practical constraints, let's say it had been effective, but you would have needed 5 times more product. Is it possible you would have just dropped this lead and focused on something else because you couldn't make enough, or what would have happened?
No, I was really early on in the purification process, so in optimizing that. So it was just luck that we had gone in with such a low dose to begin with. Maybe we wouldn't have got to that lower dose, if we hadn't had that constraint on the concentration of the product. So,
okay, so all of this is happening in your PhD. Was that 2010 to 2014? Is that right?
Yeah, well remembered.
So we now know, with the benefit of hindsight, that R21 works in kids, in humans, and it protects babies against malaria. And that took time to prove out in clinical trials. And there was, to give a spoiler to the audience, the R21 vaccine was actually approved a couple of years ago for wide use. Along that journey, I mean, you finished your PhD and you started doing other malaria research elsewhere. Did you, when you finished your PhD in 2014, did you have a feeling that, I just invented a vaccine that's going to get used by millions of people? Or did it feel more like, okay, I've proven out that this might work and I'm going to pass it on, but we'll see. Yeah, what did it feel like?
I think most people in the field were quite skeptical about R21 in the early days. They couldn't really see the point in making a vaccine that was so similar to RTSS. And the initial results, kind of backed that up. That, there was reasons for that skepticism. It didn't look very different in the preclinical work in the mice studies. There wasn't a really strong reason to think that this was going to save, a lot of lives when there was already a vaccine that was very similar.
But the dosing was very different, right?
It was. And I think this is a lesson for vaccine developers, right? At the time, everybody was quite negative about R21. But if you think about things like being able to reduce dose, making it simpler and easier to manufacture, even small increases in immunogenicity, if you add all of those benefits together, you end up with a product that's easier to deploy, a lot cheaper, and maybe easier to manufacture. So, it can really have an impact without just making a dramatically better vaccine in terms of efficacy.
It's sort of scary, though, because I think as a outsider who has never developed a vaccine, if I had seen the results from preclinical tests in animals, and the result said, well, we can't distinguish that this is better from RTSS, I probably would have killed it. So, and yet now we know that it is, much cheaper to produce and it's more manufacturable and it's longer durability. I mean, it's it's sort of terrifying. What was in the system that allowed this one to actually go forward and now it's reaching people? Is it as contingent as it sounds to me?
Yeah, I think this was all the credit goes to Adrian Hill for pushing it forward and really finding out what it could do in people, right? I think so many people would have dropped the vaccine at this point. And another difference that we haven't mentioned so far is that R21 also used a more scalable adjuvant. And I think that's quite important for the supply argument as well.
And adjuvant is another ingredient in the vaccine that strengthens its immune response.
Yeah, absolutely.
All right. And so these were different between the two vaccines. And the R21 adjuvant was cheaper to produce and easier to scale up.
Yes, absolutely. Yeah. Yeah, the adjuvant used with RTSS is a GSK adjuvant. It's also used in another product. And much more difficult to scale and much more limited supply.
I remember reading that that adjuvant comes from the tree bark of a South American tree. Is that right?
Yeah. And that comes from the bark of a Chilean, I can never pronounce the name right. Is it Chiara tree?
Does that mean that you have to get that tree to get the adjuvant? Is that why it's difficult to scale up? And how come the second one is easier to scale up if they're both from these trees?
So the supply limitation is the availability of the trees, the raw material, and also the purification process. So if you imagine you're purifying all these products from the bark of the tree or from the tree, and if you purify and throw away a lot of it away, it's going to be more expensive and harder to scale. They've improved the ways they're producing the trees. So supply is increasing and it's going to be less of a limitation moving forward. And people are also working on synthetic versions and special, I'm not sure the right word, aquaculture style growing of these trees and different methods of harvesting from them to get the components.
Is it like someone shaving parts of a tree off and then doing some other, how does this all work?
I've never seen anybody do it. That's kind of how I imagined it, taking the bark off. But other people mentioned you have to harvest most of the tree or you have to kill the tree. And that was the problem. And so I think they've changed the way that they actually harvest from the tree so they can keep growing as well in some instances. So you don't just have to, it takes 25 years to get the tree and then you cut it down.
So you're looking at your lab note, Katharine?
Yeah, it's just fascinating. I'm glad I kept really good notes. It's really interesting.
Are they dated?
Yeah.
What date are you looking at?
Troubleshooting the purification process, 23rd of the 3rd, 2011.
Whoa.
Wow. What were we doing in 2011?
Yeah, 23rd of the 3rd.
I was in school.
Oh my goodness.
I was inventing vaccines actually, Saloni, but...
You didn't tell me that.
Yeah, I try to keep it private.
So what happened on that day?
I tested a few different methods and then the result, aggregation seen in sample.
Okay. Bad.
A second purification attempt. Aim, reduce aggregation.
I love it.
Do you have any random comments in there? Do people put in computerized doodles, emoticons?
No, definitely not. Emojis didn't exist then, Saloni.
No, but emoticons did.
No.
XD.
Aggregated again. Lol. Wait, so how many experiments are you doing? Is this every day you have new entries?
Yeah.
Wow.
Well, every couple of days. So I guess I want to test something. It takes a few days to test.
Right.
And then results, aggregation seen in sample.
Ruffle. Are there images of the RTSS or R21 under the microscope? We should include one
Well, if you are up for it, us including some screenshots might be kind of fun for viewers. Oh my gosh, no,
Whoa.
sorry. We
Oh
have to
my.
include this. This
Wow.
is so cool. Whoa. Okay, great.
Wow. They're so cute.
Okay, listeners, so we are looking at two images here. On the left, it looks like spots on someone's skin, maybe measles. So talk us through the left here, Katharine. What are we looking at? Those beautiful circles. Wow.
So that's a transmission electron micrograph of negatively stained R21 particles.
Oh my gosh, they're so clear. They're so clean. They're so circular.
They're so blobby.
I mean, just to restate some of, and correct me if this is wrong, Katharine, but part of the reason why having these circles that look like viruses is so useful is that our immune system is really good at attacking circular viruses. So is that a fair statement?
Yeah, so they're easier to recognize by the immune response. Definitely, that's part of the theory behind parasite particles. What reading this has just, really made me realize how much I was reinventing the wheel. Like, if this was anybody else making it or a company making it, you'd have had an expert in purification diving into this that would have been able to. I had to do all the research myself.
Oh my gosh, wow.
And figure it out.
That's so cool.
Yeah, that's even cooler.
I did have support. There were other people in the lab that I would go to for advice. And I talked to Sarah and Adrian, but I was often figuring it out by myself, or at least that's how I remember it.
It's astonishing because I tend to, sometimes I feel myself getting skeptical about patents in particular, where the trade that society is making with inventors is that if you publish in public how you did something, then we will give you exclusivity for 20 years to do with it what you want. And sometimes I'm like, well, hold on. How much are you actually going to be able to learn from reading that? They're probably going to hide whatever they can and to get away with it. And how can you learn something from reading? You need to see someone do it. But you're telling me you literally read the patents and then you just kept plugging away until you simplified and fixed all of the stuff. I mean, that's a one-person show. And I'm like, wow. So knowledge in public is an extremely big deal.
Yeah, I think, I managed to get it to the point that we could make it and show that it works. And then there was a huge amount of work that happened by others to turn it into a GMP grade process. And even then that was, a simplified or, a shortcut version to get to a GMP product that was done by the clinical biomanufacturing facility in Oxford. They did an enormous amount of work to produce the first batch. And then Serum got involved and they used all of their knowledge and know-how to probably dramatically change the process.
So I want to pause on that, because it's, my day job is I'm a funder. And then now your day job is you're a funder, Katharine. And one thing that comes out from this story that's really interesting is that a lot of universities where a lot of knowledge is generated and science is done do not have facilities to produce the vaccines that you could take into humans safely, whereas Oxford does. Is that a fair statement? And do you think that allows for a knowledge generation loop, which is unusually productive?
Yeah, absolutely. I think that's definitely one of the, the big advantages of being in the Jenner Institute. Adrian had set up this clinical biomanufacturing facility, and that meant he could really quickly iterate. He could design something in the lab with a student, designing it, and then transfer it to the manufacturing facility and quickly produce a small batch.
Does that mean we should have more Jenner Institutes doing other can you scale that to work on more diseases? Or is it just really hard to do that?
No, I think that's the way, people should be learning from that model. I think it's not cheap to maintain a facility like that.
I have another question, which is, why is it called R21? Is that a very complicated story?
I can't remember.
Wow.
Adrian came up with a name, and I think I remember asking him, and I think he said it was a 21st century version of a repeat region vaccine. The repeat region is the CSP protein, the part of the CSP protein that's included in RTSS and R21. And that's what the R stands for in RTSS.
Well, maybe you should ask him.
Yes, right now.
Alexander, why is it called penicillin? God, I just can't remember. I can't remember.
I've got an answer.
Oh. We have an answer.
So Adrian said it's the 21st century presentation of the CSP repeat vaccine.
So your earlier answer was basically correct.
Yeah.
That's great. It's so funny to me that you thought that you had made it up, but you actually remembered it correctly. I have a similar but very different story where I thought that my earliest memory ever was made up for a while. And then I found video evidence of it being true. So my earliest ever memory is of my family going to the Grand Canyon. And at the Grand Canyon, I had a pink balloon. And the only thing I remember is that I had this pink balloon and I dropped it down the Grand Canyon. And I remember mentioning this to a bunch of people afterwards. And then eventually I was like, did that actually even happen at all? Like, that just sounds like such a crazy story. Like, why would I drop a balloon? That doesn't sound like me. Like, I was 2, but it still doesn't sound like me. I wouldn't do that. And then last year, I found a video that my dad took of this whole trip that we took to the US when I was 2. And in the video, there's a segment where I'm holding the balloon, the pink balloon. And in the background, you can hear my dad telling me to drop it down the Grand Canyon.
That is pretty much a clincher.
That is a good
I was like, wow, I finally feel so validated. But also it wasn't me. I was just listening to my dad.
There's a lot to unpack there.
I was like, why would I litter?
Yeah. Right.
That's not like me at all.
It's all adding up now.
Before we move on to the science of malaria, what happened after you invented the R21 vaccine? And what happened after you finished your PhD?
Yeah. So after the vaccine was made, we then first tested it in a mouse model of malaria. And we showed that we could protect the mice against malaria. And this worked best when you used the right adjuvant. And I did, a large number of studies in mice to really look at the dose that was needed. And that's really where my PhD ended. So once I developed the vaccine and we had shown that it worked in mice, Adrian was moving this forward into clinical trials. So I had the opportunity to stay at Oxford and continue working on the vaccine as it moved into the human clinical trials. But I'd finished my PhD, so I decided I wanted to move on and, broaden my experience and learn some new skills. I moved to work with James McCarthy in Brisbane, and he had set up a model where you could infect people with malaria. This is a challenge trial, and evaluate the efficacy of drugs. And so I was working there on those studies and also developed a new challenge model as well. I then moved on to work on projects in the field, understanding malaria transmission more fully and looking at interventions to interrupt malaria transmission. That work was mainly based in Burkina Faso and it was based out of a lab in the Netherlands.
And then how did you get from there to coefficient?
Jacob hired me.
Oh, wow.
No, I mean, it's an interesting story. I wasn't particularly looking to leave academia. I was loving the work I was doing. But obviously coefficient giving or open philanthropy at the time is a really exciting funding organization that's, quite innovative.
And what happened to the vaccine after that?
So Adrian then took the vaccine forward into clinical trials. And so there were lots of other people involved in the development of the vaccine, all the investigators in Africa, the clinical investigators in Oxford, and also the Serum Institute, who licensed the vaccine from Adrian and from Oxford and worked with the team there to carry out the phase 3 trials. And then obviously develop the final product.
I think it's sort of underappreciated just how many people work on these, like actually getting the testing and scaling up to people. It takes so many people. And it makes me think also that it's not just about funding, it's also the number of people working on things like this.
Yeah, I mean, absolutely. I think this large number of people it takes is really important for me. I just played a very small role at the beginning, and there was such an enormous number of people that made this vaccine get to the finish line and have the impact it's going to have.
I think it's time to get into the science of malaria. But before we move on, my reflection on this segment is there going to be a lot of people listening who basically science nerds who sometimes it may feel like the big things left to do in science, well, they're all behind us. That's why Saloni and Jacob talk about Gaston Ramon. We've already had all these antibiotics invented. Oh, well, so wouldn't it have been cool to live back then? And I think that's entirely the wrong orientation. There are people like Katharine Collins who in 2010 to 2014 invent entirely new vaccines that are now going to be used by millions of children. And yet, she is right here. We got to talk to her right now. And you can too. You can work on important problems, and there are many people who can benefit. And it's almost scary how sensitive to particular scientists at particular times a lot of this stuff is. And it's very inspiring too. So Katharine, thank you so much for joining us.
Great to be here.
All right. So how come it took so long to develop a malaria vaccine? And why are malaria vaccines so much harder to develop than vaccines against other diseases?
Malaria is caused by a parasite, which is quite different to a bacteria or a virus. It's a lot more complex. And the malaria parasite actually has a really complicated life cycle, and it moves through at least 3 different stages. So it's first injected into the body by the mosquito. It then travels from your skin into the liver. It develops in the liver for about 7 days and then bursts out into your red blood cells, onto your bloodstream, and it invades your red blood cells. The symptoms are caused by the parasites invading red blood cells and then destroying them. And then it does this, many times. Each cycle, it produces many more parasites. And so it invades many more red blood cells. And that's what causes the anemia. And then the parasites within the red blood cell, those infected red blood cells can adhere to different tissues, and that can cause problems for different organs as well.
I read that if just a single parasite makes it to the liver, it can cause an infection. Is that true?
Yeah, that's right. And I think that's the really tough challenge of malaria, that, you can get hundreds or thousands of sporozoites from one bite. But I think we don't think it's many thousands. And you could have some immunity that gets rid of a lot of those, but just one has to get through the defenses. And that gets into the liver. And then in the liver, they actually replicate. So it doesn't stay as one parasite. It turns into many, many merozoites, and they then burst out of the liver cell and start invading blood cells. By the time you get to the blood, you've got many parasites again, and very quickly they replicate and produce more.
That makes me think, like, with both of the vaccines the efficacy is much lower than many other vaccines. But if I think about it this way, that they're trying to prevent even just one parasite from getting to the liver, then from that perspective, it sounds like they're actually really effective at doing that, at least.
Yeah. It's a very high bar. Exactly. Yeah.
Well, that's terrifying. Just one parasite. So all of that makes it much harder than, let's say, measles or flu, which are caused by viruses, and where the vaccines are produced by killing or attenuating the virus.
So there are many reasons that it's difficult to make a vaccine against the malaria parasites. First, it's this really complex life cycle. And then also the malaria parasite has been around, since Egyptian times. So it's co-evolved with the human immune system for a really long time. It's learned how to evade our immune responses really well. So every time our immune system has managed to attack the malaria parasite or find a good way to get rid of it, it's then evolved another mechanism to keep surviving. So there are lots and lots of redundant proteins in the parasite. So you think you can block something that's important, and then it just switches on a different protein instead and uses that. So that's been one of the reasons it's been quite tricky.
So the malaria parasite has a very complicated life cycle. Does it, when it goes through these different stages, does it also change shape? What actually happens?
Yeah, it looks completely different at each stage. It starts off as what we call a sporozoite. That kind of like looks like a little eyelash when you look under the microscope. It's a curved shape. And then when it invades the red blood cell, it's called a merozoite. And this is like a round, almost cone shape. But not only does it look different, but it has different proteins on the surface. So a vaccine that works against one stage isn't necessarily going to work against another stage.
Right. And that's one thing that's hard about developing a vaccine. Like, how do you pick which protein to use in the vaccine?
Yeah, absolutely. Yeah. The approach is normally to look for a protein that's really important. So you find out something that's, probably something that's involved for an invasion or adhesion. And so people would look at knocking out different proteins and seeing whether they're critical for development. If you find something that's essential, then that's going to be a good target. And the other way is that people look for what's most abundant on the parasite or the pathogen in general. But that can often be a decoy. That's an interesting one. Like sometimes the things that are abundant on the surface are there to misdirect the immune response.
Okay, so you're taking these samples from mice and you're learning a lot, it sounds like, from mice. So firstly, thank you to our mouse brethren. And Saloni, I know that you have written a lot about, in one of my favorite pieces that I read 2 years ago was about the invention of the malaria vaccine. And you wrote about how mice were originally domesticated in some sense to be models for malaria. So how did that work?
It starts out before mice. I think there were some animal models in the late 19th century. The first animal models for malaria were birds. And the way that scientists proved that malaria was spread by mosquitoes was by using sparrows and infecting them with the blood of an infected human and seeing whether it would transmit. And I think it seemed like there were a bunch of different birds that could get infected by malaria, but they just didn't translate that well. So what would happen in humans? So people tried developing drugs, like testing new drugs in bird malaria, and they seemed to work there, but in humans they didn't. They caused various side effects. And so they were trying to find different models. There are also two other types of models used in between. One was monkeys. Monkeys are really expensive and difficult to work with. And then the other was humans, right? Because in the 1920s, if I remember right, humans were used as a model to study malaria because people with syphilis could be treated by infecting them with malaria because the bacteria doesn't survive after the fevers that are caused by malaria.
It's a bit like solving your aching thumb by chopping it off to me, but okay.
Would you rather get syphilis or malaria?
Oh, the eternal question. And my answer is I'm glad that we're in the 21st century.
Syphilis seems like it was really scary before antibiotics.
Yeah.
Especially if you got neurosyphilis.
Yeah, the bacteria would worm its way up into your brain and stick around for years or decades.
I've been to a medical museum in London called the Hunterian Museum, and they have exhibits of people who had syphilis at the time. And their skulls are filled with holes from the infection. It's very scary.
Well, that's a wonderful tangent, but I was wondering about I was wondering if we could talk more about mice.
Right. Okay, so mice. So we have the bird models that aren't great. We have the monkey models that are really expensive, and the human models, which once you could treat syphilis with antibiotics, there was no longer, an audience of people who were interested in being infected with malaria deliberately. And so the next question was, let's try to find a different animal model. Let's try to find a rodent that can be infected by malaria. And I think it took quite a while to find any rodents that were infected by malaria. And there were two researchers that finally figured it out. I think there were two Belgian researchers who were in the Congo, and they were doing tests to see which animals mosquitoes had bitten. And that ruled out various animals nearby. And it didn't rule out rodents. And so they thought maybe there's something here. And they continued trying to look for rodents that were infected by malaria. And eventually they found a thicket rat that was infected with it. And this thicket rat was infected by a different strain of malaria called Plasmodium bergae. And that seemed to be much more, that's just much easier to work with. I think at first they couldn't replicate the whole life cycle of the parasite in those thicket rats because there were certain stages of the parasite's life cycle that needed a cooler temperature. So when I was reading about this, it seemed like it took 16 years for them to work that out. It was several things. So the original research was just post-World War II. And then in the 1950s, various countries did large-scale malaria elimination programs and cut down on malaria research. And so all the people who were working on that stopped working on it, or many of them did, stopped working on it. And then only then got interested again in it during the Vietnam War. And so I think it was like partly that and partly just historical contingency of like which parasite they discovered and did they look at their own lab notes from 16 years ago to see what the temperatures were in that forest where they found the first malaria-infected rat. Goes to show how important lab notes are.
It sounds like mice can tell you something, and it took a long time to get to a model that worked in the lab. But they can't tell you everything because mice aren't humans and they might mislead you sometimes.
A mouse has never told me anything.
Speak for yourself. Well, I mean, that brings me on to a question I have, which is, are mice enough? If not, what's the next step?
Well, so you could use non-human primates, but they aren't a perfect model for malaria. There's only one that's infected, that could be infected with human malaria parasites. And so none of the models are perfect. So people don't often do a lot of non-human primate work, but they can be used to answer specific questions. And so then your next model is obviously humans.
Mm-hmm.
Mm-hmm. And that makes sense because if you want to make a vaccine for humans, you probably want to test it on humans.
Okay, so we're segueing to humans. How did that begin? What were the first experiments in humans?
So the first ones were the ones that were treatments for syphilis in the 1910s to 40s. And then when penicillin was developed, it wasn't necessary anymore. But then after that, there were various experiments in prisoners in different parts of the US, I think. And what was interesting to me when I was reading about this was that, so the prisoners were volunteering for these experiments, but some of the prisoners were not just subjects in the experiments. Some of them were actually technicians and researchers who helped in the experiments as well. And one of the most famous ones is Nathan Leopold. Do you recognize that name?
Not me.
So I know this because of a film that was inspired by his life. He was a murderer who murdered a friend, I think a classmate of his. So with a friend of his, they both kidnapped and murdered one of their classmates while they were students at the University of Chicago. And while they were serving their sentences, Nathan Leopold, one of them, enrolled in one of the malaria research studies. And after that, he then became a technician. He started doing he was actually operating research as well. And the reason that I know this is because that story inspired the Alfred Hitchcock film Rope, if you've seen that. Have you seen that?
I have not seen that.
It's a very good movie where, it's the same-ish story. They've kind of changed what actually happened. But in the film, these two students decide to kidnap and kill one of their classmates for fun, essentially. And then they store him in a, what is it called? Like a box in their apartment. And they then host a party just hours after this murder to basically show the fact that they could get away with it. And the whole film was taken in 10 shots, like 10 long segments that are just stitched together. And it's an incredible film, like very, very well made.
Wow. Okay. So,
So how does this relate to malaria? Many prisoners would have been part of this malaria research in the mid-20th century. And they contributed to our understanding of various parts of the transmission process, I think. And since then, we've continued studying malaria in humans in a type of study called a challenge trial, where you deliberately infect volunteers with mosquitoes with malaria. So I think in the past, it was many mosquitoes per person, and now it's just 5 bites. Is that right?
Yeah, I guess there was work to improve and standardize the modern challenge trials. That's where they landed on 5 mosquito bites that could reproducibly infect the volunteers.
So what happens in one of these experiments? Do you put your hand into a jar that's filled with mosquitoes, or what's it like?
No, the process is quite dull, actually. I guess you normally have to travel to somewhere where there's a facility. And because these facilities do contain mosquitoes infected with malaria, they're usually under high containment. So then the volunteers need to pass into a containment area. And then a cup is often passed through a window into that room. And they have the cup, it's like a coffee cup, and it has gauze on the top. And the mosquitoes will be inside. The lid will all be taped shut so they can't escape. And then you place your arm on top of the cup and allow the 5 mosquitoes to bite you. And so they get a few minutes for feeding, and then they'll have a look and see how many of those mosquitoes have fed on you. And they look, you can see that the body gets, swollen, it gets engorged when they're full of blood. So you can tell how many have actually blood fed. If they haven't had 5 yet, you give them a new cup with some more mosquitoes in until they get to 5 bites.
Oh,
I see.
That's probably the most disgusting coffee cup I've heard of.
Not so tasty. These days, are the people who are putting their arms out, are they mostly undergrads somewhere, or who are the volunteers?
I think it depends where you do the trials. I think in Oxford, it's typically lots of students, but other people as well. But the students are often quite willing to get involved.
Legends.
I saw a picture where it wasn't a coffee cup, but it was a cup noodle box container.
Yeah, or soup. And they're often used as soup cups. They're like or ice cream containers. They're that kind of larger size. That would be used. That's usually used to hold a lot more mosquitoes. It would have been a different study, probably.
Oh, I see.
So you, in those challenge models, you're giving some of the students or other volunteers injections of vaccine, some of them placebo, and then they're getting bitten?
Exactly. That's right. Yeah.
Okay. And then how do you figure out the truth afterwards? You're just seeing which one of them faint, or?
Okay. Once they've been challenged, you start following them up a couple of days later. So the parasites will be in the liver for 7 days. You don't have to monitor them too carefully in the beginning, but you still monitor them and check the parasites haven't got into the blood from the liver. And then you're monitoring quite closely from liver emergence. So once the parasites are entering the blood, or you're expecting them to, you can see them once or twice a day, up to twice a day. Take their blood. You can look under the microscope for the parasites, see if they've got the blood in the parasites in their blood. And you can also do molecular diagnostics as well, like PCR, to look. This is a much more sensitive method. So you can detect the parasites in the blood before they will make the people sick. And so you can treat them quite quickly. And then you'll know which volunteers have been protected and which haven't.
That's very cool. And then now there are methods that are beyond mosquito bites. People directly inject volunteers with the parasite. Is that right?
Yeah, there's a couple of models. So the other models, you would inject either cryopreserved sporozoites, that's that first stage that goes into the liver. You can inject those intravenously. You can ship those anywhere in the world to do that type of study. And then the other model is injecting blood stage parasites. And so both of these are great and they can answer different questions.
And these two vaccines, the RTSS and R21, are for the first stage. And so you'd want to be able to test it against the natural infection with the mosquito bite.
Yeah, definitely.
Saloni, I got a question for you.
Oh, what's the question?
Would you have volunteered for one of these human challenge trials?
I was thinking about this back when you asked who was volunteering in the studies, because as I have described in the first episode we did, I once volunteered for an HIV vaccine trial, phase 1 trial, and I enjoyed it. And I think I probably would. I think the difficulty is that back when I did that, I was a student and I was very bored and I didn't have anything to do in my free time anyway. And I didn't have a social life. And now I have a lot of stuff going on in my life. Like I just have a lot of work.
By the way, congratulations.
Thank you.
Me, I'm still alone, but I'll get that.
But I was thinking, like, there are lots of different types of diseases that you might do a challenge trial for, right? Like you could do one for rhinovirus or like flu or cholera or I don't know what else is that, or Shigella maybe, or something like that. And all of these sound very unappetizing to me. Like I wouldn't want to do a challenge trial for any of those. Like the flu, the respiratory ones, I'm like, that's just boring and someone else is going to do them anyway, probably. The diarrheal ones.
Like, I'm not going to do that one. It's boring. I want something harder.
Right. And then the other, like cholera and Shigella, I mean, diarrhea, I don't want, that sounds horrible. And also I feel like I'm quite small. And if I lose too much weight, there'll be none of me left. And so I can't do that one. And so what I would do is a more dangerous pathogen, I think.
Okay, nice. Is malaria dangerous?
That would make it worth it. I think so, yeah.
Okay, even though malaria...
Yeah, even though it's treatable and stuff.
It's treatable, yeah. I mean, just in case listeners are thinking about it themselves and concerned, in the diarrhea ones, they do treat you. They don't just leave you. They do give you antibiotics. But yeah, those, in order to get more of a signal, they don't treat you in, within an hour. They'll probably treat you within 12 hours or something. So it's not... you got to go through some pain to get the gain.
Well, I read that with the cholera vaccine challenge trial, some people... Actually, you know what? I'm not going to finish that sentence.
Oh,
it was about how much diarrhea. It was about how much diarrhea they had. And you know what? I don't actually want to give people that image.
That's, you know what, Saloni? I think they've got it now. So my question for malaria, Katharine, how sick do you get? Do you get sick at all, or do you get treated as soon as there's any risk of sickness? Or if you're in, what happens?
I guess the idea is that they treat you before you develop any real symptoms. So you probably start to feel a bit fluey. You may get a headache. But because they're monitoring your parasitemia so closely, the plan is to treat you before you get any really uncomfortable symptoms. By the time you get treatment, you could have some symptoms. I think people do typically get symptoms. They feel rough for a day or two. But that's hopefully the extent of it.
I think that's kind of cool. Yeah, I would do it. I mean, I haven't done it, so you always have to take with a grain of salt whatever I say now. The issue, I tried to volunteer for a trial recently and ended up getting swamped in the logistics. So your point, Saloni, that it's harder once you have a job. I've had that experience too. It also depends where you live, whether there are trials around you that are convenient that you can participate in. So it comes down to those aspects as much as anything. But malaria sounds kind of fun. If the drugs are great. If you feel a little bit sick, that will make me feel like I've done something for humanity, and then I get treated. I'm up for that. Sounds good.
And also, you get to live in a cool quarantine hotel for a bit. No,
because it's safe to walk outside if you're in England, right? There's no mosquitoes that can bite you.
So you are infected, and then they're just like, go on with your life, and you just feel sick at home instead.
Yeah. They give you a little card to
Wow.
put in your wallet, which tells people what you have and what you need to be treated with in case something happens.
Oh, really?
Okay, I'm
Yeah.
not volunteering for this then.
Especially for emergencies, like if you ended up in hospital from a car crash, they
Right.
would know that they should probably give you antimalarials now. The study's
Well,
over.
I thought you'd be in a little quarantine hotel. I thought it was like a mini vacation, but you're sick.
No, I think maybe they do it differently in different places. So it depends maybe what type of trial you're in. So for the drug trials with malaria, because they actually let you develop parasitemia to a certain level, then they treat you. And then they want to measure the clearance rate of the parasites. So they want to sample you quite quickly and frequently after treatment. So for those, they do often do inpatient for your convenience. And then they let you go once you're treated. So it could be just a couple of days.
I think I'm thinking of the respiratory virus challenge trials where because you could transmit it to someone else, they keep you in a facility.
Diarrhea as well for the Shigella one to keep you in. Yeah. I realized, Katharine, in that description, though, one of the other reasons why people might not want to do it, which is you're getting pinpricked constantly because people are taking a lot of blood samples. So that's if you want to get over needle phobia via exposure therapy, go for it because you're going to get exposed.
I feel like the way to get over the exposure therapy is supposed to give you like a mild version where it's like, oh, nothing actually happens when you get injected.
That's a good point.
That seems like it would actually scare you even more. Right?
Well, I did exposure therapy and nearly died.
Would you participate in a challenge trial?
I would love to, but I really hate needles.
Oh, there we go. There we go.
Kind of ironic for a vaccine developer. But, it's
so funny that you hate needles.
I'm terrified of getting vaccines. Like when I go to get a vaccine, I have to lie down to have the vaccine. I have to lie down with the blood taken. Otherwise I might faint. I think I have fainted in the past. That's the problem.
Wow.
Huh.
And so, as you mentioned, we take blood in these challenge trials 10 to 15 times, depending on when you get malaria. So I just couldn't cope with that, unfortunately.
We must know our limits. Okay, so how come that's not enough? Or is it enough? So let's say you've taken a malaria vaccine through human challenge trials. You ready to license it, roll it out across different countries or not? I know the answer, but I'm curious.
Well, obviously, no. I guess that's not the target population. So you still need to find out if your vaccine is going to work in the people that it's intended for, which would be children in Africa.
Well, we have some vaccines that are approved just from challenge trials, right? Like the typhoid vaccine.
Yeah, that's right. Yeah. I think when the trial isn't feasible to be done in the target population, there's good arguments to try and get that efficacy signal from a challenge trial. And then you still need to get your safety data from the target population that you're going to use the vaccine in as well.
Okay, so we've talked about the second malaria vaccine, and we are talking about the animal models and the human challenge models for vaccines in general. Saloni, since you've written about the history of malaria, I'd love to hear, if you're willing, what was the first malaria vaccine development timeline like? What were the steps there? What happened?
I love how we've gone through this backwards, like we're doing the Star Wars prequels or something.
But more entertaining.
Well, I guess it's a long story. So I think I probably want to start with a quick summary of the whole development. I first got interested in this topic when I was reading about the news of the malaria vaccine being rolled out in 2021. And I wanted to write a blog post about it. And so I was starting to write this blog post, and one of the first things that I learned was that it was actually developed in the 90s. And it really kind of shocked me. And I was just thinking, what went wrong? Like, what happened? Why it didn't only get released now if it was developed so long ago? And a lot of my interest just stemmed from that curiosity and like frustration with how things could have taken so long. The short version is that I think a lot of it is because of a lack of funding, infrastructure, and in some cases also the regulatory changes and standards that there were. But I think the broader version is really, you know, this malaria vaccine essentially came out of research from the US Army. And, in the 1950s, almost every country was affected by malaria. And there were large scale malaria eradication programs in the 1950s, mostly using insecticides like DDT. And those were very effective. Like a lot of high income countries eliminated malaria in the 1950s with DDT and other control efforts. And funding also declined. So there was no more reason for a lot of high income countries to do research on malaria. And a lot of the researchers who were previously doing it became operators and managers of this eradication program. So funding for research and development massively contracted. And that kind of stayed that way until really the 1960s, when the Vietnam War was happening and the US Army was then once again facing malaria in Southeast Asia. But by then, it was kind of drug-resistant malaria. So there was once again this need for research, trying to develop new drugs that were effective against drug-resistant malaria, but also potentially vaccines. And so some researchers started working on this program to try to develop a malaria vaccine. And their names were Ruth Nussensweig, Jerome Vanderberg, and some of their colleagues. And what they did was they first tried to build a proof of concept of the vaccine. So let's see if you can protect mice by infecting them first with, killed version of the parasite. If you protect them with this killed version of the parasite, will they then be protected from another infection? And what they found was that, yes, you could do that. And this eventually, much later, led to a different type of vaccine, which we might come back to later on. But it's not really scalable. So the other option is, let's try to develop a subunit vaccine. And from our previous episode on the history of vaccines and hepatitis B, basically the idea here is that instead of using the whole organism, the whole parasite as a vaccine, let's try to find a few components, or maybe the one component that is enough to stimulate an immune response. And so that's what they tried to find. And they sort of looked at these mice that they were protecting with this killed parasite and saw, what are they generating antibodies against? What is their immune response reacting against? And they found that clustered around a protein called the circumsporozoite protein. And that protein, it turned out, was going to be a very good candidate for a vaccine. That was going to be the main component of the vaccine. And so the initial attempts were to use that protein as the vaccine. And if you remember back from our protein subunit vaccines episode, the hepatitis B one, often just when using a single protein in a vaccine is not very effective. And the reason for that is that often when you have a whole pathogen being a vaccine, or when you're exposed to a whole pathogen, there are many things about that pathogen that can stimulate your immune response. Whereas when it's just one protein from that's often not enough for your immune system to recognize that, like, this is something that we need to react to. There are obviously lots of nuances there, but that's kind of a simplified version. And so this initial vaccine, just using that one circumsporozoite or CSP protein, was not very effective. They only managed to protect one volunteer out of 6 in their first study. And the way that you improve that is often by adding an adjuvant. So an adjuvant boosts the immune response in some way. And so that's kind of what they tried. They tried different adjuvants. They tried different formulations. And eventually they created this formulation. And I forget what each of the letters stands for. And the formulation was the RTSS vaccine. And so, Katharine, what is this formulation made of?
So the formulation is made of parts of the malaria parasite and the Hep B virus. So the R stands for the repeat region, which is from the CS protein in the malaria parasite. The T is for the T cell epitopes that are from the same protein. And the S is from the Hep B surface antigen. And this is fused to that malaria protein. And then there's the excess Hep B surface antigen I mentioned before that was needed for this to form a virus-like particle. And that's the extra S. So that makes the RTS, S.
Unfortunately, by the 90s, there was no longer interest in funding it from Vietnam War era army research anymore. And they couldn't scale up and continue that research. And so they only found this adjuvant that they ended up using in the 90s. And so at that point, we have possibly more effective vaccine. And they did another challenge trial to see how effective that would be. And this time it was more effective. It was 6 out of 7 people in the challenge trial that were protected from further infections. And again, this, a 7-person study sounds extremely small, and it is. But that was one, this is a preliminary pilot type of study. So this isn't really the real deal, but it is still much better than any other study had shown so far. So this was, a much more promising candidate for further research. And so what they did next was to do a field trial with many more participants. So they ran a field trial in the Gambia with 300 men. They vaccinated them with this candidate with a placebo, and they looked at their rates of malaria infection afterwards. And in the group that received the vaccine, they had a 34% lower rates of malaria infections over the next 4 months. So that was the first field trial, and that was done in 1998. And okay, this field trial looks, not amazingly effective, but still effective in a way that no other vaccine candidate had been up to that point. And so the next step was really, let's try to get this into real clinical trials in the population that needs these vaccines, which are children. So they started with trials in older children who were 6 to 11 years old, and then younger children who were 1 to 4 years old, and then finally infants. And this idea is called age de-escalation. Basically, you're testing at first children who are least likely to be affected by potential side effects, and then the infant group who are more vulnerable. And you sort of want to make sure that it works first in adults and then in children and then in infants. But this process took a long time, and they struggled to find funding at every stage of the process, from what I read, and also needed to themselves set up clinical trial sites across Africa. Often they were just not clinics with the expertise to run clinical trials or the equipment to do testing and things like that. And the researchers themselves had to fund some of that work. So it was a very long process. And then it eventually finished in 2015. The phase 3 trials ended at that point. And that is, I think, much longer than the research process that we often have for vaccines today.
Though luckily, I'm sure that meant it got approved in 2015.
No.
What happened then?
What happened then? Well, in 2015, the phase 3 results came in, and the European Medicines Agency said this looks safe and effective, but the World Health Organization didn't recommend it for a large-scale rollout. And they asked for more pilot studies before they would do that. And one of the reasons for that was that there were some, in post hoc analyses of the data, they found higher rates of meningitis in older children and higher rates of death in girls at 2 of the trial sites. And the World Health Organization, if I understand correctly, didn't think that those signs were causally related to the vaccine, but they wanted it to be ruled out. And so they asked for pilot studies, The pilot studies took around 4 years to find funding for and to get staffing for, and they finally launched in 2019. And then another 2 years into the pilot studies, there was enough data for the Data Safety Monitoring Board to look at the data, find no increased risk of these side effects, and then finally clear the vaccine for the World Health Organization's endorsements in October 2021. So that is 23 years after the first field trials.
The safety was one of the reasons, but that wasn't the only reason, right? There was a lack of confidence in how effective the vaccine was. So it wasn't the risk, it was about the risk benefit analysis. It was only 36% effective in those phase 3 trials, and there was this safety signal.
The RTSS vaccine was going up for EMA and WHO review in 2014, 2015. That was just about when you were finishing your PhD working on the R21 vaccine, Katharine. So what was the feeling like in the malaria research community at that time? Did people have conflicting and different views, and there was a lot of debate? Was there consensus and surprise? What was it like?
I think everybody was really hopeful that the vaccine would get approved. I think there was definitely mixed opinions in the field about whether a vaccine that showed such low efficacy in the phase 3 trial should be rolled out and would actually be effective. So I think what was really important and really key was the cost effectiveness modeling that was done with the data. And I think that was quite pivotal in that it really showed that even a poorly effective vaccine or suboptimal vaccine, most vaccines that had been used in children at the time had really high levels of efficacy, like 80, 90%. So it was a completely uncharted territory. But the modeling showed that even with this low level of efficacy, because there's so much malaria, it could have quite a dramatic impact and it would be cost effective. I think that was a bit of a surprise to everybody. We've been aiming for this really high bar. And then all of a sudden there was this realization that actually quite a suboptimal tool could make a big difference. I think that was quite important and quite pivotal.
So what did it feel like when the WHO verdict came in that more studies were needed?
I think it was really disappointing. I think it's a difficult decision that they had to make. This is really uncharted territory, a vaccine with really poor efficacy and this safety signal. So it's a tough call for them to make. But I think people at the time and now still think there could have been a better way to resolve the safety signal issue. And had they prepared better for understanding the public health impact of a vaccine that has this level of efficacy, maybe that messaging could have been different when they were trying to make the recommendations. If they understood the potential value more fully, maybe that would have shifted the approach away from a pilot implementation program that took 6 years to conclude versus doing a much more rapid safety assessment.
I guess I feel like it's a little bit surprising just because even the first field trials in 1997 and 1998 also showed fairly low efficacy, but they continued working on it. And people probably should have been prepared by that point that, hey, this, even though this isn't a very highly effective vaccine, it probably will save a lot of lives. And so it's surprising that wasn't enough to, not require another study after that.
Yeah, I think you're right. And what I've said just now is my recollection of the events at the time. So, this is a while ago and I wasn't deeply involved in the process. So perhaps there was more preparation than I was aware of. But it feels like that was the missing piece that people didn't truly, at least in the people I was talking to, didn't truly appreciate the potential impact, global health impact of this tool at the time. And there were other consequences as well to delaying the rollout. So obviously GSK would be hoping that at the end of their phase 3 trial, they can scale up manufacture and start delivering the vaccine. They probably had a factory that was making it. But now there was going to be this huge delay. What were they going to do with the facilities that were producing the vaccine? And then what was that going to mean for vaccine supply later on? So all these knock-on effects as well that, in hindsight, it's easy to think differently about what we could have done differently, but I'm not sure it was so easy at the time, to be honest.
It's also just to say the obvious, so kind of strange and messed up that the way that things currently work is that to get a scalable regulatory opinion, you end up relying on the EU and the WHO when, over time we have to get to a world where kids in Ghana rely on regulators in Ghana, not these kind of more distant bodies. Because you could totally imagine, as happened with the next vaccine, R21, that some nations want to go ahead with vaccination campaigns, even if the WHO doesn't agree, and they absolutely should. So that's another part of this story that's a bit heart-wrenching.
I feel like there's also a part of this that's like what safety standards we're used to here might be different. Like the cost benefit just might be very different. And the like willingness to take vaccine at an earlier stage might be different. But I think there's also another thing that kind of drew out the process, which was a lack of funding and a lack of infrastructure for clinical trials at the time. So especially in the early 2000s, there were very few trial sites in Africa where you could actually try to do research on the malaria vaccine at all. And it was only in the 2000s when malaria funding actually grew from various organizations like the President's Malaria Initiative in the US and UNITAID and the PATH's Malaria Vaccine Initiative that was funded by the Gates Foundation and the Global Fund. And all of these appeared only in the early 2000s. And so until then, it would have been really difficult, I think, to get enough funding to set up these trial sites and to actually do the research. So to me, when I think about it, it seems like mostly a failure of infrastructure and funding. And there are also these regulatory concerns, but if we had had that sooner, we would have been able to test many other vaccines as well.
Yeah, I agree with that. I think that's a really important point. I think we take it for granted now that actually these clinical trial facilities are so well established and the investigators there are so competent that they can run their own trials, lead their own work. That wasn't the case. I remember the stories of the people leading the RTSS work, doing the tour of the sites that existed at the time and, injecting cash into a lot of these sites to bring them up to the standards that would be needed for their product development needs. And so I think, a lot of that work done by GSK has really benefited the rest of the malaria community and other, development of other vaccines as well.
Okay, so there's infrastructure, which is key, and also funding, as you mentioned, Saloni. So let's say that there had been a big burst of funding back then that was even bigger than we saw, or how would that have happened? Were there people proposing it?
There were people proposing it, but they were proposing a different type of funding. So much of the funding that we're talking about is sort of directed at specific groups to do the research or to set up the trial sites and things like that. But in 2004, some economists, Michael Creamer and Rachel Glenister, proposed an advance market commitment, which is a different type of funding model to try to spur innovation and change the commercial incentives for vaccine development. So the general idea of why did we need philanthropy and foreign aid and all of these global health programs is that there isn't otherwise a commercial incentive to develop vaccines, or it's very small. And the reason for that is that the people who are most affected by malaria are very poor, and you can't really sell vaccines at a high price because they wouldn't be able to afford it. And so here the idea is, what if we could change those commercial incentives? And the advanced market commitments is a way of doing that. So instead of directing funding at specific research groups, it's actually a pool of funding that is only available to researchers or to manufacturers if they develop a successful vaccine in trials and they reach certain criteria. So Rachel Glenister and Michael Creamer proposed this advanced market commitment where they said we should have a commitment for around $3.2 billion in total, where you would pay per person who was vaccinated for the first 200 million people, and you would pay about $13 per person. What this does is it actually incentivizes companies to develop a vaccine and take it to the finish line, both both developing it and then also scaling it up, because they're only going to get that payout per person they immunize. So I think that is a really cool model that spurs commercial development. And it's very different from the type of funding that was actually done. But the problem was that that idea didn't actually get taken up. And there were various reasons for that. One was that at the time, people thought malaria vaccine was just too technically difficult. It was too far away to be reality. And so they didn't want to use this new funding model that was proposed for something that might not ever happen. And they thought that we have to have a lot more basic R&D before we'd have a product that could reach that standard. That also just made it more politically risky. People were like, well, we want something that will definitely, or has a much higher chance of success. We want a proof of concept that this funding model could work. And the other problem is that it's hard in a commitment like that, which is essentially this legal document that funders, in this case, they would be countries, governments, or philanthropists agreeing to pay out a vaccine that meets these standards. It's hard to decide what those standards are and what kinds of products would fit those criteria if you don't have a malaria vaccine yet and you don't have things that are late in the pipeline. And so instead of doing an advanced market commitments for malaria, there was a different advanced market commitment that was actually implemented. And that was for the pneumococcal vaccine. And so in 2009, various countries and the Gates Foundation came together to fund an advanced market commitment to develop a new pneumococcal vaccine that would target the strains that were common in Africa and South Asia. And those strains were not included in the previous pneumococcal vaccines. But there was already a proof of concept that we have these other pneumococcal vaccines. It should be fairly easy to make new ones against these strains. And it worked. So there were 3 companies, I think, that quickly developed these pneumococcal vaccines, took them through phase 3 trials, and then manufactured them in bulk to get that payout from the commitment. So I personally think, it feels like a very big missed opportunity to use it for malaria as well. But many people disagreed with that at the time.
Okay, Katharine, you were around. Do you think it would have worked back then?
I'm not sure. I think the limitations were scientific, biological. We had the problems with the adjuvants. We had lack of trial infrastructure. I'm not sure it would have made things go much faster.
Well, it's hard to say as well, because this was proposed in 2004. And so at that point, the RTSS vaccine already existed and it already had an efficacy of 30-something%. And so if you were at the time and you were trying to put this commitment together and you said, we'll fund a vaccine that reaches these standards, probably the standards that you would ask a vaccine to be developed for would be higher than the 30%, I think. And so I think in that sense, it would have been hard. What's interesting, though, is that there was another paper in 2005 where they tried to model this explicitly. So trying to model the cost effectiveness under different estimates of the efficacy, like if it was 30%, how would it be cost effective? If it was 60%, etc. And this was a paper by also Rachel Glenister, Michael Kramer, many other economists, Heidi Williams. And they found basically that it would still be cost effective to fund an advanced market commitment for a malaria vaccine, even if the efficacy was only 30%. So I think to some degree, there have been a bunch of thinking on this. But at the same time, I don't think we know what the counterfactual is, because I don't think that there are enough other commercial companies that are developing malaria vaccines now that we can compare this to. The thing that is effective about the advanced market commitments is that it gets people to enter the market that wouldn't otherwise. And so it's hard to know how that could have gone, I think.
I think that's the bit that I agree with, that, there weren't other people that far advanced in their vaccine. The only thing that has worked to date is a CSP-based vaccine. There were people working on other types of vaccines at the time, but it's not sure. It's not clear any of those would have made it either. So, or look promising enough to advance.
So that's a pull funding mechanism incentive to get to the finish line. Katharine, did it feel like more grant money in more universities and more companies, wherever, would have made the difference? So there was 100 million more every year going into malaria vaccines, or did it feel like the science and the infrastructure, you can't rush that? You actually just have to do it step by step.
That's a very good question. And actually, maybe I'm going to change my opinion slightly in that, there were with more funding available, there were other people doing great science at the time. So Simon Draper at the University of Oxford has developed a blood stage vaccine called RH5. And that work was happening at the same time on the lab bench next to me when I was doing R21. But that was seriously underfunded. So people had to prioritize what they were going to support with their funding. There was limited funding. And so, the focus went on to CSP-based vaccines. And that's what the Gates Foundation funded, the RTSS vaccine, through MVI. And the Gates Foundation didn't fund blood stage. There was only one funder at the time that was supporting that work, or one main funder, that was USAID. And so had there been more money, that work could have been accelerated. And, that's shown now in a phase 2 trial that it gives good levels of efficacy, not as high as R21 and RTSS, but there's potential to combine that with R21. And so that work could have been accelerated a lot quicker had there been more funding around.
So just playing that out. So that result you just referenced, that was from about a year ago, was it? No, it's pretty recent, right? Yeah. And so if that funding had happened a decade earlier, could that result have come about a decade earlier, do you think? Or that's just a funding question, really?
Yeah, I think the product was the one they were working on in the beginning.
No!
Wow. Oh, that is incredibly depressing.
I know. But I think, credit has to go to Simon Draper and the funders that supported him. Lorraine Soissons at USAID, they kept that work going with quite limited budgets, and they made it to the finish line. Well, not to the finish line, but to the clinical trials now and the efficacy trials.
Thank you, Simon. Thank you, Lorraine, for believing against consensus and keeping it going.
I have a question related to this because both these are different stages of the vaccine. And so I'm wondering if you had a vaccine that combined, the vaccines against both of these different stages, would you expect that to have a higher efficacy? And have people tried to do things like that?
Yeah, I think that was the original approach from Adrian when I was making R21. He also had a liver stage vaccine. The other vaccine that he had developed was the ME-TRAP vaccine, and that targeted the infected hepatocytes. His theory was that if you could reduce the number of sporozoites that get to the liver, then the liver stage vaccine would have an easier time at finding and clearing out those infected liver cells. And that was tested, and it didn't work. So that was, a great theory that didn't work. Now people are looking at combining the CSP-based vaccines with the blood stage vaccines that Simon Draper's developed and others. There are lots of other people working on blood stage vaccines now as well. But the preclinical models are really difficult to evaluate all stages of the parasites in the same model and the combination of those vaccines. So we really have to get into humans. And that's the work that Simon and Angela and his team are working on at the moment.
Wow. And so could that have also been sped up by 10 years?
Yeah, that vaccine that they're now putting into clinical trials with R21 was being developed at the same time as I was developing R21. So, that could have gone quicker.
This is crazy.
It wasn't exactly hard to spot. It was right next to you on the lab bench next to you. I'm going to cry.
Jeez.
I mean, so as someone who works in philanthropic funding, I think the main lesson here is just like that funding in global health R&D is so tight that you have to make these prioritization decisions that are vicious and impossible. And so a more sane society would have more long-term government and philanthropic funding that was less correlated, more decision makers, more money total, so that you could get more shots on goal. And so, I think, for anyone listening who is wondering what the solution is, I think that's the most obvious general solution staring us in the face. And so I don't want to hide it. There's obviously all these specifics we can get into that are more technical, but that one is obvious. So we just have to say it out loud. Sorry.
Jeez.
No, I agree with that. I don't disagree with the decisions that were made about what you had to prioritize at the time. Like, I think they were tough decisions that people had to make. But had there been more money, we could have moved things a lot faster.
I feel like what's even more depressing about this is that, like, despite all of that, like, malaria is still one of the more well-funded diseases in global health world. And there are various others that are, large burdens and get tiny fraction of the funding.
I am wondering if we could just talk about the two vaccines already approved next to each other, and then what's coming next after these two, what needs to be done next in the invention pipeline. So RTSS versus R21. I will tee this off by saying, I don't know that there are different comparisons you can make, but one fact just to state up front is that neither has faced a rollout where tens of millions of children have received the vaccine. So perhaps the biggest problem is simply that there is not enough reach of either of them, let alone comparing between the two. That said, what do we know about the data that has come in about how they look? So there's more data coming in every year, but what do we know so far about efficacy, about safety, and other factors? Katharine.
The headline efficacy figures from the phase 3 trials look quite different for the two vaccines. So RTSS, we mentioned it was something like 36% efficacy overall. And actually for the population that was used for the phase 3 trials for R21, that was most similar, the efficacy I think was 57% overall. And then for R21, the phase 3 result was something around 75%. I can't remember the exact numbers. So this looks like R21 may be a lot better. But there's some nuance and detail about how these trials were conducted and where they were conducted and when they were conducted. That means we probably can't directly compare these trial results. So I don't think you can claim that R21 definitely has a high level of efficacy. There were different things happening at the time. So different other malaria interventions that were being used, like bed nets or seasonal chemo prevention, drugs that were given to children every year. So those results aren't directly comparable. So I think we have to try not to make that comparison unless we run a head-to-head trial. But what I think is interesting about the R21 data that's starting to emerge is that it does look like it may be more durable than RTSS. So the decline of protection seems to be slightly slower. So it may, even if the efficacy isn't much different, that peak protection they get, it may last a little bit longer. So that could make quite a big difference.
I see. So, but in both cases, if your first shot, you'll probably get at 5 months old. Is that about right? And then you might get another. The ideal is you get one at 5, one at 6, one at 7 months. And you're saying that the immune response from R21 is lasting for longer after those first shots?
That's right. There's actually a fourth dose that's given around 18 months.
I think my takeaway from the difference between the two is mostly the manufacturing and the cost. Is that also how you see it? And are there any other differences? Why is there such a huge difference in the cost of both of these? And is that because of the dosing that we talked about at the start?
I guess the other big differences are the scalability of the vaccines. So R21 it seems to be easier and cheaper to produce. And that may be in part due to the lower dose, and maybe also new technologies that have been used to produce the vaccine itself. So newer yeast expression systems and things like that. And also the adjuvant that's added to the vaccine. So we mentioned earlier that the adjuvant was really important for the RTSS vaccine. We found the same with R21. Only when we used a certain adjuvant did we see protection. And that the adjuvant that's been chosen for R21 was Matrix-M. And that doesn't have the same supply limitations as AS01, and it's slightly cheaper as well. So that makes it more scalable.
Yeah, I mean, it's funny. It's not the property you'd think about first, scalability and how that might have implications for cost and price. But last price that I saw for RTSS was about €9 a dose, and there are 4 doses. And then the last price I saw for R21 was, the original price was $3.90, and it maybe has come down to $3. So we're talking about a 3x, say, difference, which may not sound like much, but is decisive in a lot of these cost effectiveness calculations, because global health funding, there's just not so much of it going around. So, when you think of cost effectiveness, it's not just how effective are these vaccines, it's how much it costs to actually deliver them to the kids who need them. So anything you can do to bring the numbers down it could change the equation.
Yeah, and just going back to the delivery schedule you mentioned earlier, Jacob, which is giving doses of the vaccine at 5, 6, and 7 months. And then there's this booster dose or final dose that's given around a year later. And none of these time points are actually aligned with the schedule for other infant vaccines. So what this means in reality is that children and their parents and caregivers have to travel for each of these visits at another time. And what we're finding is that people are not showing up for all of their doses of vaccine. And so if they're not getting the full course, it's going to reduce the effectiveness of both of these vaccines.
Right. If you have to travel 3 months in a row, but you're, busy taking care of your other kids, or you're at work, you can see how you might miss a dose. And then if that happens, then whatever we saw in the clinical trial may not reflect the reality of what kids are getting protected. So more work to do there. Okay, so those are 2 vaccines, and they're imperfect, but they are helpful for kids. Do you think they're good enough, Katharine?
If we did this again today, could we make better vaccines?
Yeah, I think, oh gosh, wow. That's a tough one. I mean,
scientifically, not in terms of the funding or trials or anything like that.
Yeah, I think we have better techniques and technologies for developing vaccines. Structured guided design, reverse vaccinology, computational protein design, things that you've talked about in your previous podcast episodes, where we really try and understand more specifically what type of immune response you want to generate. So you can take the whole protein that you think is important and show that to the immune system, but it might generate a mixture of helpful responses and unhelpful responses. And so using these more sophisticated techniques, we can try and understand which parts are really helpful and design the proteins or the antigens to really elicit those responses more carefully and more specifically. And so using those techniques, people are already doing this. They're working on trying to make better CSP vaccines, better blood stage vaccines, and also vaccines that target the transmission blocking stage as well.
But why were you hesitant before?
It's not clear that any of those will actually be any better. So I think there was a really interesting paper published by the Protein Design Institute by Neil King, someone that we support and think is an incredible institute for using computational design to really improve vaccine antigens. And they tried to make better versions of R21. And I can't remember what the paper showed exactly now, but there were many candidates that they assessed, and they used this incredible methodology to try and improve the vaccines, but none of them looked substantially better than R21.
What do you think of the whole sporozoite vaccines, where it's just irradiating the whole thing and injecting that as a whole? That seems like, in earlier research, seems like it was pretty effective.
Yeah, absolutely. I think those were some of the first data that really stimulated the vaccine development community. They showed really early on, decades ago, that you could protect people by using these irradiated sporozoites. And that work continued. So a company called Malaria with Steve Hoffman and others developed the PFSPC vaccine. And this worked really well with 3 doses in people in the US, so people who had no experience with malaria infections before. But when they ultimately got this into children in Africa who had malaria infections in the past, the vaccines didn't work very well. So that hasn't moved forward for that population. There's actually been some really interesting work more recently but they've used genetically attenuated parasites. So instead of irradiating the parasites and then using them, they've made them so that they can't survive beyond the liver stage. But importantly, if these parasites live to the end of the liver stage, then they are really protective. And there was a recent study where they showed that a single dose of this vaccine provided really high levels of protection in a challenge trial. But again, this was in people that haven't been exposed to malaria before. So while this looks really exciting at the moment, it still needs to be tested in the field in people who have been exposed to malaria and then ultimately in children as well. But it's really exciting. It's pretty game-changing if you can get a single dose vaccine to protect against malaria. We've never seen that before. But the challenge with these parasites is that they need to be stored in liquid nitrogen. So you can't just kill them like we have done in the past with other whole pathogen vaccines. They need to be viable. They need to be able to survive in the liver. And that's going to be quite challenging for delivery in Africa because there aren't liquid nitrogen facilities everywhere.
Why do they have to be so cold?
That's the way of putting them in stasis.
Or is it just preserving them?
Preserving them, yeah, so that they can be woken up. They've either got to be kept alive until you inject them, or you can try and cryopreserve them in a way that they can be thawed, and then they'd still be viable.
What if someone found a different way to preserve them for a long time? Are there other options? Can you freeze frame a vaccine?
No.
I have no idea. I've been watching a lot of Pokemon, and Team Rocket has all of these crazy tools and equipment to do things. And I feel like they would have a laser beam that could just freeze someone in place.
That would be cool. Yeah.
Let's take a note. Let's look into that. I think that could be pretty interesting.
And I know a panda that's just started there.
Yeah. Let me just. Actually, I've already got too much in my laser beam column. Someone else is going to have to fund that one. But
also, this laser beam thing that could freeze could be used much more widely than just malaria research.
You could zap people miles away. It's interesting I don't hear people talking about this more, given how important it could be.
Yeah.
Now, on our current technologies, boring. So it sounds like my sort of takeaways from this section are that, when people talk about vaccines, the thing you always hear about is efficacy. COVID vaccine is 90% efficacious, blah, 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 blah. But that is one number. Firstly, that number does not bake in to the public narrative duration. It does not matter if something is 90% efficacious and lasts for 3 months, if it then decays. Secondly, it doesn't bake in cost. What's the manufacturability of this? How widespread is it going to be in a global health context? But then in addition, it doesn't bake in how many doses do you need? And in a context where people might not be, they're juggling a lot, may not come back for a second, third, or fourth dose, getting down from 4 to 3, 3 to 2, or 2 to 1 is just an incredibly big deal, not for scientists, but for people in the real world who are actually going to be taking these vaccines. So, Katharine, is that a fair summary?
Yeah, absolutely spot on.
It reminds me of, the HPV vaccine, where it was originally a 3-dose vaccine. And then it turned out that actually 1 dose was really effective. But it took a really long time for them to change the recommendation on how many doses people needed. And now that they have that new recommendation that you only need 1 dose, you can actually scale it up much more widely than before.
So, Katharine, if you had to make a guess, I'm going to put you on the spot. In 10 years, will we have an approved vaccine in use, which is, say, 2 doses for kids?
10 years, I think 2 doses is too high a bar. I think we can get to 3. I think we can get to a more durable and a more protective vaccine with 3 doses.
What are the barriers to making it happen?
I don't know. Maybe I want to change my answer there. I think 10 years, I would put it 60% chance we can get to 2 doses.
Okay. But it's all to play for, you're saying. It really could go
Yeah,
either
it's
way.
not a done deal. I think 60% chance we can get to 2 doses, like 90% chance we're going to have a better vaccine.
Okay.
3 doses. 90%, that's too
I
high.
see.
80.
Okay. So you're saying, we've got to plug away and get to a better vaccine than even the one you invented.
Definitely. R21 is suboptimal. I mean, it can have impact like we've discussed, but it's not a game-changing vaccine. The delivery challenges, the durability, that all needs to be solved.
Okay. So challenge number 1 then. So you're saying there's a good chance if we really push for it, we can get to a better vaccine. Maybe it's 3 doses, maybe it's longer duration. But we have to also be aiming at the same time for even further improvement for kids. And that's hopefully getting to perhaps 2 doses, perhaps, transmission blocking, that kind of thing.
Yeah, absolutely.
So it's all to play for. Okay. Saloni, how are you feeling at the end of this section?
This makes me sad. I feel like it's so sad that, one, our technology has improved so much, but it doesn't seem to have made that much of a difference in terms of the efficacy, except for this one vaccine, which is really hard to scale up because you need to cryopreserve this parasite stage, which means that it sort of rules it out being used widely. And the third thing is that there were like various vaccines that are in late stages of trials right now that could have been in late stages 10 years ago. It's just like the combination of all of that is just incredibly depressing to me.
Yeah. Well, can I try and cheer you up?
Yes.
Just to reorient to how many lives a vaccine can save that is even imperfect. If R21 or RTSS, R21 seems more likely now, could scale up to more children who need it, tens of thousands of children's lives would be saved already. And then Katharine is telling us that within 10 years, we have a good shot at an improved vaccine that's even better. So, that is going to matter to a lot of children. And science, the reality of this problem is the hardness of it is set by nature. And nature is a vicious test setter sometimes. The fact we've got this far, that is pretty impressive. And we have a ways to go, but there's line of sight to improvement, even if not to the absolute 100% blocking 1 dose thing.
I'm happy for, the children who are getting these vaccines. But it's just that counterfactual. It's just very hard to get out of your head.
Yeah. So Katharine, as you look to the next 10 years, how do you feel?
I'm optimistic. I think we can do better. I think we've got, great people doing incredible work, great new tools. And more people thinking about the problem end to end. So more people thinking more than just efficacy, which, all the other criteria you raised that we should be considering. More developers are now aware of all of those things to consider in the development pathway. And so I think there's a lot of hope.
There's also the current rollout, right? That we could scale up.
Yeah, I think that's a tough one. I think it's a really difficult funding environment. So we don't have enough donor support to provide R21 and RTSS to all the countries that would like it, would need it, and are requesting from Gavi. So if we had more support coming from the different donor countries.
The UK is cutting back, US cutting back, Japan's cutting back.
Germany as well. What I read from a report talking about the situation at the time 2 years ago, back then, both of the vaccines had been pre-qualified by the World Health Organization, and they were being rolled out. And they estimated that it would take another 12 years after that for all children under 3 in the countries with high malaria prevalence to be vaccinated. And another 2.5 million children were expected to die being unvaccinated from malaria. And that the main constraints to increasing the number of children who were vaccinated fast was a lack of funding. And I remember reading this and finding it surprising in one sense that this was the only constraints. But also, I think after everything that we talked about, thinking that it was actually not that surprising and just very depressing. But the fact that if you had a few more $1 billion of funding for Gavi, which supports vaccination in countries around the world, you could vaccinate enough children to save another 300,000 lives. The other thing that was interesting to me about this was that one of the reasons that there was this funding constraint was that one of the countries with the highest burdens of malaria was Nigeria. And Nigeria had just increased its GDP enough to be placed in a higher threshold of like a higher income country, according to Gavi. And that meant that they were no longer eligible for financial support to purchase these vaccines. And that meant that a lot of children would go unvaccinated and that the cost was just so much higher after just passing this threshold. And it seemed very bad to me that was the case. But there is some good news, which is that there was another deal where the price of R21 was reduced from $4 to $3 per dose. And that would save around $90 million and help vaccinate another 7 million children.
So all in all, what do you each wish that donors and funders and other decision makers understood about vaccine development that they're getting wrong?
I mean, I think it's one of these situations where actually throwing money at the problem would make a difference. And that is something that people might find surprising in other fields. But here, like funding Gavi would actually go a pretty long way, or funding the other types of malaria vaccine research would go a long way. I think that's quite surprising to people.
I think we should really be learning from our experience with RTSS and R21. And the biggest problem with both those vaccines is getting all of the doses into the children that they need. And so thinking more carefully about how you develop a product that's deliverable earlier on in development could have such a dramatic impact on, the impact the vaccine can actually have. And I don't think the researchers doing the work, the ones running the very first trials in humans, are thinking about that enough.
Right. It's less pizzazzy, but actually testing different delivery schedules in the clinical trials can have these incredible downstream effects of what gets recommended for millions of kids.
And maybe it's not fair to put that all on the researchers and the developers, actually. I think, they're asking for funding to do the work. Funding's tight and limited. Funders will give you the bare minimum to do the bare minimum. And so you can only test one schedule. So you have to go with, what you have the data on already, or what already looks promising, instead of exploring what the other options could be. So maybe that's also on the funders to really understand that as well.
I think another thing is that people may not know is just how important it is to have clinical trial infrastructure, to have better ways of running trials more efficiently, but also just more sites, more people doing these tests, being able to test multiple vaccines and not having the situation where people have to prioritize funding for one vaccine over the other at the expense of testing the others as well.
But I think there's also another part to the story as well, in that with malaria, everybody knows about malaria. The burden's really well known. So as soon as a malaria vaccine was developed, countries were asking for it. They knew they had a problem. They wanted the vaccine and they're pushing to roll it out to protect people. So those vaccines can have an impact. But there are other vaccines that we've tried to develop in the past where we didn't have good data on the burden in all the countries that possibly needed it. And so I think the Hib vaccine was a good example. It was developed, it was ready to be used, but countries didn't know they had a problem. It was this huge delay in rollout. So there's so much more that needs to be done when you're thinking about developing a vaccine beyond just developing the product. thinking about how it's going to reach people and how people are going to understand whether they need the vaccine, what the demand is.
That was quite an episode. That is our first episode where we had a expert interviewee with us the entire time. Thank you so much, Katharine. And now it's time to conclude with what are some of the things we learned or that stuck out to us from this episode? And I'm going to start. Number one, how much you can learn from patents. It turns out that there are people who read them and then they tweak them and then they change them and then they make new inventions. I mean, that really gave me hope for public knowledge again.
That was really cool to hear about.
Saloni, what stuck with you?
So I had a bunch of thoughts. The first one, which I was thinking about before we started recording, was that, and this is a very unpopular opinion, but some PhDs are just better than others. And some fields are better than others too. And I know it's controversial, but I think more students should go into infectious diseases and should go into vaccine development. And I have many thoughts about which fields should move into those, but I won't name them. They'll probably... They'll know who they are. The other was that I thought it was really interesting to hear about the Jenner Institute and how it was set up in such a way that there was a manufacturing facility that researchers could work with, and they could learn from that stage of the process in manufacturing. And I thought maybe more institutes should be set up like this, where you can see the, both the basic research and the translation and the manufacturing happening in the same place and learn from these different stages. That I thought was really a cool thing to learn about. The other that stuck with me throughout this episode was that malaria is very complicated, especially compared to the other pathogens that we've talked about in previous episodes. It has many different stages of its life cycle. It changes shape. It's harder to develop vaccines for malaria than for many other diseases. And there were various parts of that process that were just very tricky to do scientifically. It was hard to find good animal models that helped replicate what the disease would be like in humans. The next was that a lot of the research funding for malaria was affected by priorities that high-income countries had. So in the 1950s, during the global malaria eradication program, the funding for research dried up. And a lot of researchers were made program operators in the eradication program. And that stalled research until the Vietnam War, where the US Army troops were, facing potentially drug-resistant malaria. And there was now a renewed need for research into new malaria drugs, but also malaria vaccines. And that is where the RTSS vaccine originally came from, was research from people who were working on that during the Vietnam War. So that stood out to me as well. That we sort of think of research happening, in the lab, but it's really actually influenced by all of these much broader considerations and the, historical context and things like that I think people don't appreciate enough.
So the first one for me is probably the cost. Scalability and deliverability are actually really important. People should think about them earlier in the process. I think, it's clear from the R21 and RTSS experiences that the vaccines could have much greater impact if those things were considered earlier.
I also thought that just within that process, it's not just the broader concerns, but like at every stage of vaccine development, these things are really important. Like how you develop the adjuvants and how expensive those are, or how that affects the efficacy of the vaccines and how accessible they might be later on as well. And then things like how does the dosing of the vaccine that's being developed affect how easy it is to scale it up across countries or like across children, millions of children, I think is something that people underappreciate. And then similarly, the vaccination schedules, the fact that the malaria vaccine is taken at different ages than other childhood vaccines, and how that affects uptake of the vaccines is something that was new to me. And just this idea of making things more efficient at all of these stages could be really important. I've been reading the book, The Origins of Efficiency by Brian Potter. And one of the key things that stood out to me from that book was just how much progress we've made in, medicine, but like engineering and all parts of life from improving the efficiency of things that people have already discovered. And that stage can often make the difference between something that is possible versus something that's actually used by millions of people. And I think that's actually a huge part of the picture. The other thing that I have always been thinking about is just how different things could have been. Like what other alternative universe we could have lived in if things were different. And the key things that I think about here are, one, how things would be different if there was more funding. And second, how different things would be if there was better infrastructure for running clinical trials. And we talked about a bunch of examples of how that could have been different. So one that probably would have sped up the development of the RTSS vaccine, the first malaria vaccine, having the clinical trials sites set up earlier, that would have made a difference. Having more funding, that would have made a difference. But then we also talked about other vaccine candidates for malaria and how more funding for that research could have changed the picture and sped up the trials for those vaccines. And then finally, the rollout of the malaria vaccines that have already been approved could be sped up with more funding. And that funding was the constraint and still is the constraint to getting that out to more children. And I think that often people think of this as purely a scientific problem, but I think that's not the case. And that in many situations, when we're talking about diseases that affect people in poorer countries, often commercial incentives and funding and, the historical context and all of these things actually have a very big impact.
I think it's really easy to look back and think about how things could have been done differently with hindsight. But it's really important to remember that these were really uncharted times for this type of vaccine. And it's really quite amazing what was achieved over the time. Obviously, we all, wish things could have been done faster and can keep going faster in the future. But this is quite remarkable what was achieved. But I think it's important that we do look back. I think we should all be looking at the experiences of the past and learning from them so we can improve what we're doing as we're developing future vaccines, learn from those mistakes. And now as a funder, I'm trying to take a lot of those lessons and those experiences and apply them to the development of next-gen malaria vaccines and strep A vaccines and any other products we end up working on at Coefficient Giving.
What you just mentioned made me realize that the alternative universe that we could have lived in, it could have been faster, but it also could have been slower. And I hadn't thought about that.
I think my main takeaway from looking backwards into the past today is about the present and about the future. And it's that we live at a time of scientific wonder. People who invented world-changing technology are among us today. Invention is often a process of building on other people's work, tinkering in the lab, and taking care of those lab notebooks, looking in an electron microscope, trying an experiment, changing what you started with, trying another experiment. And these people are heroes. They're real people. Sometimes you can even get them to come on your podcast. Thank you very much, Katharine. And to anyone listening, you may know one of these people, and society may not recognize it yet, and they're still inventing and they're still trying stuff. You may become one of these people in the future. And when it comes to science, we are all in this together. We're trying to figure out what is true and what we should do next. So I just want to end by saying, Katharine, thank you very much. This was very enjoyable, and I'm very excited about everything that comes next.
Thank you. It's been great to be here. Great to chat.
If you enjoyed this episode, You should rate us on Spotify or Apple or wherever you listen to this and share it with everyone you know, including any parasites that you are currently infected with.
Maybe they are heroes too. You never know. They could contribute in their own way. Please share this one in particular with anyone who's considering starting a PhD.
Yes.
Great. See you next time, everyone.
Bye.
Bye.
Bye.